good afternoon. I think it's afternoon everywhere across the country now. Thank you very much, Tina, for getting us started. We also want to start by acknowledging and respectfully acknowledging that we are calling in from the traditional territories of the Musqueam, Squamish, and Tsleil-Waututh First Nations, and very grateful for being able to do so. My name is Mayank Singal, and as Tina said, I'm the lead for enteric pathogens at BC-CDC. And joining me is Dr. Natalie Prystajecky from the Public Health Lab. And we're going to be talking to you about talking to you about norovirus, how it has been evolving, and some of the public health impacts that we have seen both in BC but across Canada and also in other parts of the world, and how we use some of the novel surveillance tools to enhance our outbreak response. So to give you an overview of what we can expect, and I'm just trying to advance my slides. There we go. Okay. Is to just give a brief overview of the epidemiology of the norovirus epidemics that many of us probably come across in our professional lives, but perhaps also our personal lives as well. Norovirus is a very common enteric pathogen, and because it's a very common enteric pathogen, there is special role for wastewater surveillance. As you know, that's with a lot of the other enterics pathogens, we do have case-based reporting to public health, but that's not necessarily practical for a bug like norovirus. So how can wastewater fill in some of those surveillance gaps? We'll go over some of that. We'll talk a little bit about how the bug has evolved over the last couple of decades and what impact it has had on the severity of the epidemic that we see from year to year. We'll go over a particular outbreak response. And this is when the norovirus got into our food supply chain and how we respond to that here in BC, and then also just going over some of those outbreaks and surveillance tools that have been relatively newly used here in BC. So I'll get it started in terms of just the basics of what norovirus illness is. And the way I always talk about norovirus is talking about my own experience in my family. A couple of years ago, when it was a Saturday morning, I was actually watching my two boys. I was solo parenting that particular day, and my older son comes up to me and says, "Papa, my tummy hurts." And I was like, "Well, okay, we'll watch and see how things go." And 15 minutes later, all of his breakfast was out on the floor of our living room. So it is something that hits you very hard. My son, he probably vomited several times within the next 2 hours. So it tends to be very intense vomiting when this happens, and it really starts very quickly. It's not something that builds up over time. Oftentimes it'll start with vomiting and then kind of transition into diarrhea. Some people can have a little bit of fever and abdominal pain. The main concern with this virus is typically dehydration. People can get dehydrated just because of the amount of vomiting and diarrhea that can occur. The good thing is that it's mostly a self-resolving illness in most people. Gets better after 2 to 3 days, but it can certainly cause a lot of morbidity because people are extremely weak with all that dehydration. So it can definitely knock people out in terms of being able to attend school and work and do the things that they typically would be able to do. Most of the transmission happens person to person. Occasionally, we can see foodborne illness outbreaks, and we'll go over one of those with you in a short while. So what's been going on with norovirus more recently? So on your left, what you're seeing in the blue curve, what you're seeing is the wastewater activity for norovirus, or wastewater detection of norovirus in a local wastewater plant from in January 2021 till February 2025. And you can see those sort of cyclical peaks that happen every year. It happened sort of end of 2021, end of 2022, and then later in the last year's peak was a little bit delayed. It was actually in 2024, and then we saw another peak. We see another peak that's not quite peaked yet, but late February of this year. So what seems to be apparent from the blue curve is that we haven't quite peaked yet, and we're already past the previous peak. So something's going on, as in it seems to be that we're having a worse season for norovirus this year. And we've seen similar media reports across the country as well in a number of other Canadian provinces as well. On the right side, what you're seeing is a curve for a number of outbreaks that have been reported to the CDC in the United States. In the red curve is the number of outbreaks that have happened between August and July of the various years. This particular year has been very active, as you can see in the red curve, compared to the previous years that are visible that are shown in the shaded areas. So something's going on this year that's making the public health burden of illness much higher than previous years. So what could be going on? So when we think about this from an epidemiological perspective, we have various hypotheses. We think about, is this a new virus? Is it somehow changing? The virus is changing, so that's a new strain, or somehow there's some mutation that causes a bit of immune escape. Are there some changes in the population, like waning immunity? We haven't seen this virus for a long time. Maybe something special about the post-pandemic state, where in general we saw less circulation of typical pathogens in our day-to-day lives. Potential environmental factors like travel, or long-standing changes related to climate change. And we always have to be mindful of our artifactual causes. So these are basically these are causes that are not true increases, but maybe more related to things like more testing or more reporting, increased awareness, or better lab tests. So those could also be happening. So we think one of the reasons, at least one of the big reasons why we've seen this, is an association with the circulating genotype. So I'm going to show you a little bit about the genotypes, and then we'll show you some BC specific data as well. And Natalie will go over what the different genotypes are as well. So on the top left, what you're seeing is on the left side of the slide, what you're seeing is the different genotypes over the years for the United States, as well as a number of European countries. So over in the red, the G217 seems to be going up. That started to go up in 2022, 2023, and then it has been going up since then. Whereas the G24, which had been the predominant strain since about 2012 worldwide, is starting to come down. So what you're seeing here is that last year being 2020, 2023 and 2024 in the US, for example, about 30% of the genotypes were G217. And this year, which is 24-25, we're seeing that jump from 30% to almost 80%. So a big jump. So that is correlating quite nicely with this figure that you're seeing in terms of increased number of outbreaks. So there's at least an association between the shifting of the genotype and an association with greater burden of illness across the population. And you might be aware of some of the more recent news articles that have been coming across, coming across these articles in many different parts of the world, both in the U.S., Canada, and in Europe as well. So it seems to be something that's affecting a number of different geographies across the world. So I'm just gonna pause here, and I'm gonna shift. Hand it over to Natalie. Natalie will go over a little bit more details about the virology of the virus and how that impacts us here in public health.
Great. So so you may have seen on the previous slides the mention of some of the genotypes type names. You'll hear G2.4, G2.17. So I will give you a little crash course in norovirus nomenclature and a little bit more detail about the virus itself. And so noroviruses is a broad group of viruses. They belong to the Calliciviridae. And what you can see on this part of your slide on the left-hand side is that you'll see a number of different notations going, G1 through G5 up to G8, and you'll see that there are different species associated with them. And what you'll note is that G1 and G2 are the ones that are associated with human. You'll see that under G2 there is also swine, but the swine G2s do not infect humans. And in fact, this is one of the most important details about understanding norovirus transmission is that the strains that are infectious to humans are only infectious to humans. So if you do have norovirus, if you have a norovirus infection, you have at some point been exposed to human fecal material. And so we are not expecting that animals, that this is not a zoonosis. It is a zoonotic viral group because, you can see canine there, you can see swine there, but the human-specific strains only infect other humans. And so, when we are thinking about how we've acquired the infection, it is not through manure, unless that manure contains human waste. And so these are the most common modes of transmission, is that it is transmitted through the fecal oral route. And one of the most notable features of norovirus is that it has an extremely high viral load. So when an individual has diarrhea or they vomit, there's so much virus in there that it can be almost impossible to save yourself from it. So we do see a very high rate of secondary attack rates within households. And I tell my own personal story of my children having norovirus, where my child, became ill. I just happened to have gloves and a mask nearby. So, as I was taking care of them in the bathroom, I did mask and glove and, got them all cleaned up and settled. But what I didn't do is mask and glove when I was scrubbing the carpet because they had something red before dinner or with dinner. And our beige carpets had a big red stain. The stain actually never came out. We ended up replacing with hardwood floors. And I also developed norovirus. And so, it is just so contagious. And often when caring for small children with norovirus, it's very hard not to get it. And you can imagine that, if an individual is sick in a public washroom or they haven't washed their hands properly or they're sick, at a daycare or in a long-term care facility, that the infection can occur quite quickly. So it's contamination directly or indirectly through the fecal oral route. So whether or not you're caring for someone who's vomited nearby and there's particles in the air, or it could be through indirect contact. So touching surfaces that become contaminated, shaking hands that may have not been washed properly. But I think the big take home is when you see norovirus infection, it has been associated with a human infection prior to that. Next slide. All right, so when we talk about norovirus genotyping, you may, historically, we used to just sequence just the capsid region. And so I'm showing that there. It's in the open reading frame number 2. And what actually happened is that generally what happens with norovirus is we see something called strain replacement. So every couple of years, a new strain will emerge and you'll see sort of an epidemic of increased activity. And so it's, at this point, there comes a time where most of the population has been exposed to a certain strain of norovirus, and eventually another strain can now emerge and escape the immune response and then become the most dominant strain. So we saw instances where we saw infection rates that were consistent with strain replacement, but when we genotyped it, it showed that it was still the same strain, G2.4. But some clever individuals figured out was that when they sequenced further out away from the capsid, and they also sequenced the polymerase, they actually found that although it looked like it was one strain at the capsid, it looked like a different strain in the polymerase. So recombination had actually occurred. So this is what we do now here in Canada, as well as in the United States, is that we sequence the capsid and the polymerase so we can capture genetic recombination of this virus. So I'll go through in the next slide how we do the naming. So what we have is that we will sequence both a little bit of the capsid and a little bit of the polymerase, and then we will type both of them. And then we compare that sequence to a reference database and assign a genotype name. So you'll see that you can have something where in the capsid, sequence is consistent with G217, and at the polymerase, exact same strain, G217. Now, there's also the possibility that there's something different at the polymerase and capsid, and these are the recombinants. So you'll see G22, so that represents what's going on in the capsid, and a P16. So it represents a G216 in the polymerase. And likewise, the strain that was most common until this year was a G24, so G24 at the capsid, and a P16 at the polymerase. So this is the way that we name them, and I'll talk a little bit more about what patterns we've seen here in BC using this genotyping approach. If we go to the next slide. So I will start by saying, as a disclosure, is that the BC CDC does a lot of testing and sequencing of human stool and vomit to support the province of BC. The caveat is that the testing that we do is available not only at our site, but it's also available at frontline labs as well. And in fact, there was a newer panel of testing, the infectious diarrheal panel, that was introduced over the last few years in the province, where more testing can now occur in the front line. So we do support some reference services, so confirming different testing. We will also receive samples for genotyping specifically or sequencing. And we do support, particularly here in the Lower Mainland, testing of specific facility outbreaks. But we do not have a centralized database for all norovirus data in the province. So it's a subset of this data. But it is this is a valuable source of data to show us what's happening in the province. So the first thing to note is that you can see that there are waves. So there is definitely some seasonality, and we see more activity in the winter months. So this is for this reason that historically norovirus was called the winter vomiting illness. And the other thing that you will notice is that it's predominantly G2. We very, very rarely see G1s in the subset of samples that we collect. If you go to the next slide. And we also, so here at our site, we sequence one representative sample from each outbreak, as well as a handful. So, roughly, depending on the year, between 10 and 50% of the non-outbreak samples. We do this so that we can see what, the genotypes that are predominant in outbreaks versus non-outbreaks. And so what you can see is that there has been, over the sequential years, a shift from predominantly yellow, which is G24 Sydney, and a shift towards seeing more G217 P17. And so we are not like what Mayank showed in the States, where they're at about 80% G217. We are still seeing a predominance of G217 P17 over G24 P16, but we haven't seen such a huge shift. So we are over 50% G217, but we are not nearly. We do still see this split. The other thing to note is that this season that we have right now, which is a very big season here in the province, as you can also see in the wastewater. This is not the first time we've seen G217. We actually did see G217 in 2022. We saw a number of samples, but it wasn't able to outcompete other circulating strains. If you go to the next slide. So one of the things, as I mentioned, is that it was just a subset of data that I was able to present to you, and that's because there's no centralized requirement to that all norovirus data be captured in a centralized way. Norovirus outbreaks are reportable, but individual cases are not reportable. We also have a decentralized testing model, and we only sequence a subset of samples. The other thing is that outbreaks can be declared and managed without lab confirmation. And so they can be declared using something called the Kaplan criteria. And then the other thing is that we know that it's very difficult to compel patients to produce a stool specimen. I will say that, in my, when I described my norovirus case, my own family's norovirus case, I will admit that was not lab confirmed because, one of the things was that I did have in my kids' bathroom a stool collection kit. I was ready for this, but I was so sick that I was not even going from my bathroom to their bathroom to go and get it. And so that just sort of describes the sort of situation people are in. They're often so sick that they cannot necessarily seek out getting a sample collected. And then all of a sudden they're better. And that is one of the things with norovirus is that it's quite a big illness before a very short period of time. And so we often see this in outbreak settings where individuals are feeling better. They might be chatting to one another that maybe they all had, individuals may have gone to a restaurant and all felt ill. And then by the time they've actually realized that they might have had an infection, they're no longer able to produce a stool sample of quality for a norovirus investigation. The other thing is that our testing guidelines here in BC actually exclude norovirus as if norovirus is the cause of illness with a really rapid onset and resolution, it is not in line with our diarrheal testing guidelines, where we say that you shouldn't test unless you've had diarrhea for over 7 days, unless you're in a very specific criteria, which is that you're part of an outbreak or you're clinically vulnerable. Mike, if you just forward, the one thing is that wastewater surveillance can address a lot of the surveillance gaps. And so this is something we've been doing since the beginning of the pandemic. If you go to the next slide. So many of you may be aware of BC's wastewater surveillance program and may have become aware of it starting in 2020, 2021, when our data became more available. But my journey with wastewater actually began in 2018, where we started to test for it with input in collaboration with Metro Vancouver. We started testing for enteric viruses in wastewater. Metro Vancouver was interested in understanding why, how much virus is incoming and outgoing from the wastewater plant. And us as a lab that does work in development, we wanted to A, develop a good method for wastewater, and B, to understand how much norovirus is coming into a wastewater plant from the community. So we had been starting to do this work early in 2018. But when the pandemic hit, this work was put on pause and we focused our efforts on detecting enteric virus or respiratory viruses instead. If you go to the next slide. One of the things is that we leveraged research methods that we already had for those enteric viruses. And so, the tools were the same. We had to concentrate the virus, we had to extract the nucleic acid, and we had to develop a qPCR to be able to enumerate. If you just go forward one, Mayank. One of the things that's really important also when we're doing any of this work is the controls. So you have to have positive controls, negative controls, as well as inhibition controls. So wastewater in itself is the material there can be very difficult to amplify. There's a lot of heme in the wastewater, which can inhibit the PCR steps, but we can check whether or not there's inhibition by spiking in a known quantity of another target. And in fact, our inhibition control was already linked in to our norovirus assay. So we just kept running the norovirus assay as part of our inhibition control. So we had this bonus data dating back to the beginning of the pandemic where we had norovirus data in wastewater. So if you go forward. So with this, we had with this bonus data, and with this, we could develop this historical trending of wastewater data. And so you can see that, again, we have that seasonality, just like we had in those outbreaks, and we can see the peaks and the troughs, and this has been really valuable data for us to start to understand this, to better understand norovirus throughout the province, not just in the Lower Mainland, where the majority of our samples were coming from for the clinical assays. If you go to the next slide. We can use norovirus to fill in some of our surveillance gaps. And so this is some new data, right? Just, very, very fresh data where we've been looking at norovirus hospitalizations against norovirus concentrations. And you can see that the trending works very, very nicely. The next slide. And we can also just look at different parts of the province and look to see that, one of these sites actually doesn't have that cyclical trend that other sites do. And we're just, this is very interesting to understand how to use this data to, better understand norovirus trends in the province and potentially any interventions that need to be done as a result. Next slide. And so one of the things that we notice, and if you just click forward twice, Mayank, there, oh, just once there, okay, oh, perfect, is that we know that some of these specific troughs or peaks, to be honest, these peaks are associated with very specific events. So if you look at the first peak that I've highlighted there, actually there's two peaks. These peaks are associated with a time where there were outbreaks in oysters, as well as a high amount of community transmission. And also what Mayank was saying is that, we are continuing on this upward trend. We might be just starting to see the top of the mountain right now, but we have seen concentrations higher than we've ever seen before in wastewater, and this seems to be associated with G217. So we can use the wastewater data to corroborate information that we are maybe seeing in an outbreak setting or in the community setting with respect to strain replacement as well as outbreaks. And so with that, I'll pass it back to Mayank.
Mayank, I think you're muted.
Oh, thank you, Tina, and thanks, Natalie. So in association with increased community transmission, we can also see outbreaks related to norovirus. So these can be foodborne illnesses as well. So I'll go over this one, our most recent example, and what you're seeing is an epidemiological curve of an outbreak that we managed a few months ago, that started in November 2024 and was declared over in January, about three months ago. It totaled about 124 cases, and that spanned 54 clusters. So clusters are basically people who got sick together, essentially. We received these reports from our regional health authorities here at BCCDC, and we assess them. We look for things like, are there any alternate causes of illness? Like, was a restaurant inspection done? Was there sick food handler there when the people were dining, and whether there was somebody else sick at home that could have potentially passed that illness to the case. If we rule out all those causes, then there's a good chance that the food was the cause of the norovirus illness. And what we do with these is that we'll pass these on to our federal partners, with the CFIA, Canadian Food Inspection Agency, which then collects this data for further action. And we'll go over what those actions are in a moment. But I really want to highlight that this is really a team sport. A lot of different service lines here at BCCDC come together. Canadian Food Inspection Agency has a very central role, as well as Department of Fisheries and Oceans. And that's a federal department. Environmental and Climate Change Canada, Health Canada, and Public Health Agency. We all basically come together. So a lot of the work that I'm really presenting here is really a team effort. So what do we do when we come across these types of outbreaks? So one of the first things for us is to make sure that there's public awareness related to this. And some of you may have come across these news items back in middle of December. About increase in GI illness, noro-like illness, following the consumption of raw oysters from BC, and part of that is so that people are aware of what's circulating, and then they can make decisions that are in alignment with their values. So that's one piece. The other big piece is around closure of harvest sites. So this is a map. I'm gonna orient you because I know that a lot of people are not from BC. So you're looking at an area that's about 100 kilometers northwest of Vancouver. The light gray area on the left side of the screen is Vancouver Island. That's where Victoria, our capital, is located. And there's a smaller island called Denman Island. Between these two islands, there's a body of water called Bain Sound. It's about 30 kilometers in length. So 40% of oysters that are harvested in BC come from Bain Sound. So it's a very, very active area for harvesting of oysters. And what you're seeing here, all those red rectangles that you're seeing is where these sites were actually closed. So when we get multiple reports of oysters from these sites that are causing illnesses, then our federal partners will go ahead and close those sites for harvesting. And so that's one of the major public health actions that comes out of our investigation. The third piece around public health action is advisory. So BC CDC, CFI at the federal level, and also FDA in the US will issue an advisory to restaurants to not serve these oysters. And then here locally in Canada, the Canadian Food Inspection Agency has that role in terms of to recall those oysters as well. And BC CDC also issues an advisory to its restaurants here locally as well. So those are the sort of big three actions that come together in terms of advising the public, those closures at the harvesting sites, and then recalls and advisory to restaurants. Those are the big public health actions. So we've seen these types of scenarios before, and Natalie will go over a little bit more about what we can do about these scenarios.
Right. So one of the things that you'll notice here is that these outbreaks have occurred multiple times. So you'll see, three sort of newsworthy outbreaks that are associated with oysters here in BC. And so, these oyster outbreaks continue to occur. It's not every year. And one of the things that's quite interesting is that, we assume that these let's just start with, how do oysters become contaminated? They become contaminated through contaminated water. And that water becomes contaminated specifically with human waste if they do specifically have norovirus. And so we know that that oysters are filter feeders, and they can filter a very large volume of water and bioconcentrate contaminants that are in the water. But of course, there's contaminants more than just norovirus in water. So why are noroviruses so much more associated with oysters than other pathogens? And it's because there are ligands within the gastrointestinal tract of the oyster that are specific for norovirus. It's just one of those unfortunate fates of biology that oysters specifically bioaccumulate norovirus. And so when we see that there's often pollution in our marine waters, you would think that there would always be contamination of norovirus if these oysters, but that doesn't happen. It seems to be sporadic. And I think that one of the things we need to understand from a research perspective is, how do we know, what are the risk factors that are associated with norovirus contamination of oysters? If we go to the next slide, we'll go forward to, I guess, this is the question of what can we do about this? And so, as I mentioned before, when you see a norovirus contaminated oyster, it has come in contact with human waste. And what are some of these human waste sources? If you just put the arrow forward one, Mayank, this slide demonstrates many different sources of fecal contamination, but certain ones have no bearing or at no risk to shellfish contamination because they are from non-human sources. So pets, runoff from parking lots, runoff from livestock, those are not going to be a risk for human health, nor are seagulls or sea lions or other marine mammals. What the sources are things like failing septic systems, discharges from marine vessels, and discharges from wastewater plants, as well as swimmers and other sources of contamination. If we go to the next slide. This is really what we're trying. We do have a project, and it's called Gemstone, and it is funded by Genome British Columbia with partners from various ministries, as well as Malahat Nation, First Nation, as well as BC Shellfish Growers Association, to better understand why is it that sometimes of the year we see, or sometimes of any given year, we have oyster outbreaks, but sometimes we don't. So first thing we know is that oyster outbreaks tend to be associated with heavy rainfall, periods of time when there's low sunlight, periods of time when there's low surface temperatures, as well as downwelling. So this really allows the sort of recirculation of deposited materials from the lower from the lower depths of the ocean. We also know that norovirus tends to, these times where there's heavy rainfall, low sunlight, are associated with times where there's more norovirus contamination or more norovirus transmission in the community as well. And so if you go to the next slide, one of the things that we can do is we know that norovirus wastewater is a signal of how much activity there is in the community, which is related to how much might be deposited into the ocean environment during contamination events. So we can add in these models all these different features along with concentration of norovirus in wastewater, that bonus data that I described, and that can help inform the outbreak risk as well as preventative action. Next slide. So as I mentioned, Gemstone is a project where we're looking into using these different data sources together. So we're looking at not only understanding the norovirus risks, but also understanding other sources of contamination as well and where they might originate from. If you go to the next slide. It's really focused on a premise of trying to understand who pooped in, not necessarily the river, but in this instant, the marine environment where oysters are raised. And we're using specific genetic markers to better understand the sources of pollution, whether they're human and non-human, and if they are either of those, where they might be originating from, to better understand the risks to the oyster production, as well as to reduce the impacts on human health. We go to the next slide. So this is just a high-level overview of what the project intends to do. And one of the things that we're really focused on is working with our partners and identifying the best sites to do this research to make sure it's most impactful to them, but also developing tests that could be used by the Shellfish Growers Association or by Malahat Nation or other involved in oyster production, so they can have tools available to do this type of assessment with laboratory methods. Next slide. Back to you, Mayank. You need to unmute Mayank.
Oh, yes, thank you. Rookie mistake. So yeah, no public health talk would be complete without talking a little bit about prevention. So what's really remarkable about norovirus is that we really strongly recommend washing hands with soap and water. This is one area where sanitizer is not considered adequate. It's just not strong enough for norovirus. Norovirus is a very hardy virus. It can survive in the environment for a while, so it just needs the full soap and water treatment. For people who are sick, they should stay home until 48 hours after resolution. I think that's a fairly common public health recommendation for this type of an illness. And then on the environmental health cleaning side, it's really important to use a solution that can kill norovirus. A lot of the bleach-based solutions are quite effective at that. And here in BC, especially, or wherever there's a lot of shellfish consumption, we recommend cooking the oysters during periods of high transmission, because there might be a bit more circulation happening. To flip it to the organizational perspective, we need policies to detect and reduce those transmissions in facilities, whether that's care facilities or healthcare facilities, schools, daycares, all those places. Hand washing stations again need to be made available, because sanitizer is not enough in this particular state. Apologize, I don't know if you can see my teams pop up, but okay, we'll carry on. Facilitating sick individuals to be able to stay home. That's one of the things that happened here in BC, is that when people are sick, there is some paid leave, provincially legislated paid leave for folks, and of course. One area that's near and dear to many people on this call, probably around outbreaks, policies around case isolation and enhanced cleaning in the environment, and ensuring any activities that carry on in those facilities are safe for the scenario. Alrighty, so I think that concludes most of the talk, most of the material that we wanted to share. We do want to acknowledge the many different people and groups that have contributed to this work. It truly is a team effort. Both on the public health side as well as on the labs and our partners outside of BC CDC as well.